in the BioSENS lab under Jonas Limson and Ronan Vogel. My research entails a lot of the use and study of biosensors. Now, a biosensor is a device that incorporates a biological component in order for it to be able to specifically detect a target of interest. Now, these biological components is known as a biorecognition agent. And the best example of a popular biosensor would be the blood glucose monitor, which utilizes the glucose oxidase enzyme as its biorecognition agent. Now, in our lab, we focus on the production of synthetic oligonucleotides known as aptamers. And when these aptamers or biologically sourced components are incorporated into a sensor, that's known as aptasensors. Now, for this to become an aptasensor, we need to immobilize the aptamer onto a transducer surface. The transducer surface basically amplifies the signal or the binding event, and this can be done by numerous procedures or principles such as optical, piezoelectrical, colorimetric, or electrochemical. So obviously the transducer surface ultimately determines the mechanism of detection. Now, once we've actually fabricated these sensors, so chosen our transducer surface, found our aptamer, we analyze or characterize certain properties of the sensor that we've just made. So 3 important factors that we consider are the sensitivity of that aptasensor, which is basically how well it can detect the target of interest or how little of it. The specificity of the sensor, which is how well the sensor can differentiate between molecules that are of similar structure or shape as the molecule of interest. And then finally, for a real-world application sensor, we would have to incorporate both of these and be selective, which is, will there be preferential binding to the target of interest in a complex matrix or medium?